So today I want to give a general overview of what we do at Stalicla, and I have also asked Laura to join me to present very recent results that we have that we are getting ready to share as prior to publication in this symposium, further characterizing the biological validity of our this subgroup of patients called phenotype 1, for whom we are entering a first-in-human phase 1b study at Cincinnati Children's Hospital with our STP1 compound. I want to give you a little overview about Stalicla. So Stalicla is a clinical stage company developing a first-in-class systems biology and machine learning platform with a goal to advance precision medicine for patients with neurodevelopmental disorders, including ASD, ADHD, schizophrenia. Stalicla has a twofold asset structure. Its DPI v3 platform is a proprietary technology specifically developed to address the challenges of NDD drug development, parsing the heterogeneity through the integration of non-behavioral clinical data with multi-omics and pharmacological data. The company is also fueling its own pipeline with a primary focus on advancing its internal pipeline with its first program currently entering clinical trials, as I just mentioned, and a secondary focus on establishing NDD precision medicine technology strategic alliances. So overall, Stalicla's approach is novel and leading the way of NDD precision medicine. Our platform has validated its proof of concept through ASD Phenotype 1 STP1 drug package. That is the first validated subgroup of patients issued from the platform and the corresponding precision drug. So ASD and other neurodevelopmental disorders are mostly behaviorally based diagnosis. They are underlined by significant heterogeneity in genetic background and clinical presentations, and they co-occur at rates higher than would be expected by chance. So overall, on this basis, 40 years of research have generated vast amounts of data, but these data have not been integrated consistently, specifically because of this behavioral-based approach underlying the diagnosis. So key players in the field are still pursuing a one-size-fits-all approach, and up to date, this has resulted in failures in clinical trials, further supporting the fact that there is a need to stratify homogeneous subpopulations, find homogeneous subpopulations to make precision medicine a reality for patients living with ASD. So Laura is going to dive into our platform technology that has enabled to identify this first subpopulation corresponding drug.
Mm-hmm. So in this slide, you can see an overview of the main pillars and key assets that integrate the DPI platform, which, as Lynn mentioned, is our core technology that we use for developing precision medicine in NDDs. And basically, we leverage on proprietary NDD tailored knowledge bases, as well as to the access to genetic and clinical data from large-scale NDD cohorts that we get from our unique and growing partnerships, in order to apply a first-in-class integrative systems biology and multi-omics approach in the NDD space. So one of the key processes that DPI platform enables is the identification of non-behavioral biomarkers characterizing NDD patients, which is what we call the endophenotyping. And then these non-behavioral biomarkers can be used then to cluster the patients using machine learning and being able to identify more homogeneous and clinically actionable subgroups of patients. And on the other hand, DPI uses network endopharmacology to find fast-track drug candidates that can target specific subgroups of NDD patients. And basically, overall, this platform and the key modules that integrate it allow us to match biologically driven patient profiles with drug profiles. And these are actually the two possible entry points in our platform. So we can either start characterizing patient profiles to then try to find the drug candidates that can target those in an efficient way, or we can start characterizing drug profiles, drug candidates, to then identify profiles characterizing patients that are likely going to respond to these drug candidates. And in summary, DPI allow us to reach Stalicla's main goal of matching the right patients with the right drug treatments.
So the first output of the DPI platform is Stalicla's third therapeutic package, which includes the patient population and the corresponding drugs. So our first patient population, ASD Phenotype 1, is a non-behaviorally characterized population with association of clinical sets of signs and symptoms that were identified by our platform, a corresponding drug treatment, which includes compound drugs that have never been approved on the same market with excellent safety profile, potentially enabling to capture a significant portion of the ASD Phenotype 1 population. STP1's MOA integrates a PDE inhibitor with anti-inflammatory properties and an NKCC1 inhibitor, and STP1 is specifically tailored to the pathophysiology of Phenotype 1. In parallel, Stalicla is developing for Phenotype 1 and STP1 a companion diagnostic leveraging on the discovery of its Phenotype 1 specific biomarkers and advancing the companion diagnostic development for STP1. So the first validation of ASD Phenotype 1 was conducted based on defined association patterns of non-behavioral clinical sets of signs and symptoms identified by DPI and verified in an observational clinical trial conducted in 2017 at the Greenwood Genetics Center in a cohort of 313 patients where the DPI criteria were applied for a review of medical records and patient medical interviews. And this observational trial confirmed the existence of this subgroup of patients and the association at a higher rate than would be expected by chance of these specific clinical sets of signs and symptoms and the prevalence predicted by DPI. So this subpopulation is referred to as ASD Phenotype 1. The Stalicla research group went further to validate this population at the biological level with the first metabolic validation. So the cell lines, lymphoblastoid cell lines from patients with Phenotype 1 were compared for their metabolic profile with control LCLs and non-Phenotype 1, so autism non-Phenotype 1 lymphoblastoid cell lines. And we were able to establish a distinctive metabolic profile fingerprint for ASD Phenotype 1 cells compared to non-Phenotype 1 and controls. And these specific conditions allow the identification of ASD Phenotype 1 with high sensitivity and specificity, providing a first validation of the biological validity of ASD Phenotype 1. So this is the first validation of an idiopathic, non-monogenetic group of patients with ASD. Additionally, this specific metabolic response was also associated with a specific pathophysiology, since specific metabolic response consistent with the molecular model characterized by ASD, by DPI for ASD Phenotype 1 as overactivation of the NRF2 pathway was confirmed through further metabolic assays, showing that non-Phenotype 1 and controls, when exposed to NRF2 activator, were phenomimicking Phenotype 1, suggesting a saturation or an upregulation of NRF2 as the driving pathophysiological mechanism in Phenotype 1. So overall, Phenotype 1 is characterized by dysregulation of cyclic AMP that's predicted to be central to ASD phenotype 1. Low levels of cyclic AMP are expected to induce a variety of pathological cellular responses related to cellular growth, cell proliferation, and immune response, all characteristics of ASD phenotype 1 in the observational clinical trials. Treating patients with ASD phenotype 1 with a PDE inhibitor is predicted to increase cyclic AMP signaling in patients and correct the core cellular dysfunction associated with a disease. However, long-term exposure to cyclic AMP results in dysregulation of intracellular chloride mechanisms, justifying the association of AKCC1 antagonists, which will maintain the efficacy of the driving therapeutic PD inhibition over time. So overall, the STP1 is therapeutically tailored to the pathophysiology of ASD phenotype 1, to the validated pathophysiology of ASD phenotype 1. So my screen, yes, we also were able through further assays to confirm the involvement of cyclic AMP as the core pathological mechanisms in ASD phenotype 1. ASD phenotype 1 cells actually show a distinct biological profile and a distinct drug response to cyclic AMP, to exposure to cyclic AMP, compared to ASD non-phenotype 1 and control cells, which is quite obvious in this pharmacological curve. So in cells with ASD phenotype 1, STP1 PDE inhibitor mimics the increase of cyclic AMP concentration and induces similar cellular energy production, showing the STP1 PDE inhibitor compound is increasing cyclic AMP in the same way as shown in the previous assay. So a further validation of ASD phenotype 1 was conducted through additional assays, and Lara is going to comment the further biological results that we obtained on this population.
Mm-hmm. Yeah, so we basically wanted to replicate the first biological validation of ASD phenotype 1 that we had obtained using this metabolic screening from patient-derived cell lines using other high-throughput omics technologies. And in particular, in this slide, you can see results that we obtain from metabolomic profiling from patients' plasma. So as you can see here, this assay again show a distinct profile for ASD phenotype 1 patients compared to controls, thus further validating the biological identity of ASD phenotype 1. Also importantly, this analysis allow us to identify specific metabolites that are dysregulated in ASD phenotype 1 patients, and that opens the possibility to develop a companion diagnostic tool. So we were then able to further validate the existence of this convergent pathophysiology in ASD phenotype 1 by conducting gene expression profiling. So in this slide, you can see results from principal component analysis of gene expression profiles from ASD phenotype 1 patients, ASD non-phenotype 1 patients, and controls. And as you can see here, we were able again to observe a clear separation of ASD phenotype 1 patients compared to other idiopathic non-phenotype 1 patients and controls. And interestingly, some of the molecular alterations that we found to be specific to ASD phenotype 1 don't segregate ASD non-phenotype 1 patients from controls, which is another example of the existence of a high heterogeneity in ASD.
So overall, our platform has identified ASD phenotype 1 through the aggregation of specific clinical sets of signs and symptoms. And these criteria have been used and applied in an observational clinical study involving idiopathic ASD patients, and have allowed to confirm the existence of a specific subpopulation matching these criteria with an association of criteria occurring at a higher rate than would be expected by chance in 25% of the enrolled population. So DPI has also allowed to generate a probabilistic model linking the pathophysiology of ASD phenotype 1 to the dysregulation of specific molecular pathways, including NRF2 and cyclic AMP. And further, the analysis of patient biosamples have confirmed highly significant biological signatures for these patients, opening the way to cost-effective candidate biomarkers and the development of a companion diagnostic in the clinical trials for STP1 addressing phenotype 1 patients. So they have also provided the evidence of the possibility of developing precision medicine-based treatment in polygenic conditions such as ASD phenotype 1. So overall, this is the first precision treatment package advanced for a biologically validated subpopulation of patients with ASD, and the corresponding treatment STP1 is entering first in human studies. So we are now initiating a phase 1b randomized placebo-controlled 2-dose ascending study enrolling 24 adult patients with ASD phenotype 1 at Cincinnati Children's Hospital. The primary objectives of the study are safety and tolerability and pharmacokinetics, and we are also including innovative exploratory outcome measures of efficacy, including EEG and novel cognitive batteries that had previously been integrated in recent Fragile X studies, successful Fragile X studies. Overall, this is the first interventional trial with ASD phenotype 1 patients only. So overall, in 3.5 years, Stalicla has achieved key milestone, including the identification of ASD phenotype 1, as well as 2 additional subgroups of patients with ASD. We have entered 3 programs into our proprietary pipeline. We have brought our first ASD phenotype 1 specific STP1 pipeline into clinical trials, and we have incorporated CP3/5 collaborative pipelines, all corresponding in our portfolio, all corresponding to clinical stage drugs for which identification of responder profiles and must advance these drugs. So these are third-party drugs. So we've also secured significant financing for the company and are standing as a leader in the field of precision medicine for neurodevelopmental disorders. Additionally, and probably most importantly, Stalicla is a profoundly patient-centric company with a group of scientists who have both a strong drive to develop translational solutions for the NDD community, as well as a strong personal involvement in making this objective succeed.
There have been a few questions. So even though ASD phenotype 1 is biologically defined as a subgroup, have you seen any shared clinical or symptoms and can you share any details on that?
Yes, absolutely. So in our observational trial, we also conducted behavioral characterization of ASD phenotype 1 patients. Overall, they are a quite heterogeneous subgroup, but they do share common characteristics, including hyperactivity. They do tend to present with cognitive impairment with a mean IQ of 63. This is interesting because the hyperactivity feature resembles a key characteristic of Fragile X, in the underlying pathophysiology, which is linked to cyclic AMP abnormalities, is also involved in Fragile X.
That's really interesting. So a technical question, what platform was used for your gene expression profiling of ASD phenotype 1?
Laura? You're muted.
Yes, I believe we use the NovaSeq, the new Illumina platform for sequencing for transcriptomics.
question asked, why is the study only 2 weeks? Could the treatment be effective in 2 weeks? I assume they mean in as little as 2 weeks.
Well, this is a safety tolerability study. Obviously from a regulatory and a toxicology perspective, you have limitations in the time that you can pursue a study. Nevertheless, based on the known mechanism of action that we have involving PD inhibition, we could expect, or we could hope for early surrogate or predictive signs of efficacy in EEG markers or in anchored battery endpoints specifically related to processing speed or reaction time. The reason why we're mentioning this is because in healthy volunteers and in elderly subjects, PD inhibition can induce within a matter of hours changes in reaction time and processing speed. And in behavioral studies in autism, early predictors of social communication, changes in social communication endpoints have actually been established as, reaction time has actually been established as an early predictor of changes in social communication endpoints. So we are integrating exploratory endpoints, including EEG, that can allow us to capture these early changes.
A biomarker to a biomarker, right?
Absolutely.
Yeah, that's really interesting. I didn't know about that. Well, wonderful. Thank you guys from coming. I know you won the frequent flyer award last year. I'm sure, guessing it's probably
well,
pretty.
This year I probably lost all my points.
It's probably pretty late where you guys are, but thank you so much for attending.
We're in Barcelona, but we're here and happy to answer additional questions. Very good questions and interesting questions. Thank you for those who asked them.
Actually, there's one more that popped up as since you're defining your target group biologically, how do you choose your clinical assessment tools? That's a good question.
This is also a very good question. There is a relationship between establishing biomarkers are not only serving the purpose of patient identification, they also serve the characterization of the pathophysiology of the patients, and having a better understanding of the gene expression profile of our patients, we can evaluate how these genes or these markers are expressed in the CNS and look or parse for more specific outcome measures.